Okay. Hello, can you hear me okay? Yep, good. All right, hello everyone. So welcome to the week 8 lecture. So we've got a guest for you today. Most of you already know Hayden from the practicals. Also, he was one of our tutors in LQB385 last year. He's got quite an interactive presentation for you today on analysis and visualization of genomics data. So he's currently doing his PhD. He's already got a master's and an honours before that, so he's quite experienced in a lot of these things, particularly in bioinformatics. So he's going to talk about that.
Amazing. Thank you, Dan. And thank you, everybody, for turning up on a Tuesday afternoon. I understand 4 o'clock's a bit of an ask, so I do appreciate everybody still turning up today. There's a bit of a competition going on, if you couldn't tell from up the front. There's going to be like a Kahoot-style quiz as we go along, and whoever has the quickest answers and the most correct answers will get, in order of winning, a first place Lindor chocolate, second place Cadbury block, and third place twirl. So hopefully, would you mind joining us in the front section here? It's all right. So hopefully we get some good stuff going out of it. So we're going to be covering 3 different sections today, the first being a little bit of philosophy of science, looking into how we actually think about the significance of our results and what the implications of that are. Then looking at different types of genomic data and how we analyze it afterwards at the end. So moving on. To start with, this is just a bit of an example question to get you all involved. What is my favorite color? Red, green, or blue? So this is just a chance for you to scan. If you want to participate, to go ahead and scan the QR code and join in. Oh? Interesting. You do not have to do this, but there is chocolate on the line if you feel so inclined. Oh man, red, green, or blue. What a choice. Anybody else still trying to join, or are we all good? Anybody else looking to join still? All right, we're going to go forward to show results. And you picked blue, probably because of my shirt, but I kind of tricked you all. None of them are correct. My favorite color is yellow. So that one was because it didn't count. All right, but moving straight on into the content. If we're talking about the philosophy of science, of course, we go back to these old guys. We've got Plato and, oh my goodness, what's his name? I forgot his name. Socrates. Sorry, thank you. So thank God Socrates had the idea of looking out into the world, because Plato, for some reason, thought that you could just think about things really hard and come up with the ideal version of something and find out knowledge about it that way. Not so with Socrates. So thank God he came up with 2 types of reasoning. The first being deductive reasoning, meaning that we have these first principles, these knowable, immutable facts about the world, and then you can derive knowledge from them. The first meaning that if all humans are mortal, and then Socrates is a human, therefore Socrates is mortal. And then the other idea he came up with is essentially going in the reverse, where we have observed knowledge and then we extract the first principles from them, inducing the general principles and then deducing the general principles. So for the example of inductive reasoning, I mean, if all the swans I ever see are white, then all swans are probably white. But there's a little bit of a problem with inductive reasoning, because how many swans do I have to see before I know for certain that all swans are white? I have a question for you. This one also doesn't count, but it's just to see what you think about the general state of sciences. Do you think that science uses more inductive reasoning, meaning that it looks at the knowledge and measures the world and then creates general principles from that? Or do you think science uses more deductive reasoning, meaning taking established principles and then deducing knowledge from that? Give it a guess. It should be like a percentage, like you have 100 points to distribute, so you can sort of pick which one you're thinking. No harm, no foul. If you have no idea, that's okay. More inductive or deductive? All right. Anybody else still going? All right, let's run it. What did we think? Wow, exactly, pretty much 50-50. I was not expecting that. But that's pretty cool. If you were to ask me, I would say that science is pretty dominated by inductive reasoning, meaning that we look and we measure the world and then we come to generalized principles about that. I would say where deductive reasoning is mainly used is more in mathematics and a little bit in physics. However, if science is, or at least the realms that we work in, are very inductive, what does that mean about how much we have to observe things before we can create our general principles? This leads us to this guy, Dr. Karl Popper, who pretty much came up with the whole problem with the way that science is run now. And he put to the world, how many ravens do you need to see before all ravens are black? And what he was essentially getting at was this idea of the inductive leap, which is how many real measurements do you have to do before you're willing to step off that whole, is it going to be real or is it not, and create the hard and fast general principle. I want to put another question to you later on. I mixed myself up a little bit. Now, scientists have this guy, Sir Ronald A. Fisher, to thank for a lot. There's also a whole lot of controversy around this guy's whole career about having to go into hiding and using pen names and like student t-tests and a whole bunch of stuff like that. But thanks to him, we have this wonderful thing called the p-value, which basically governs all of scientific work nowadays. Now, I want to ask you lot, as fledgling scientists, some of you may be, what is a p-value? Now, this one does count, by the way. So we've got, what does a p-value represent? It represents how important an observation is, the likelihood of the observation occurring randomly, or statistically how important the observation is. Oh, that's interesting. I think I might have messed up here on this one, sorry, because I messed up on that one. I apologize. The answer is, sorry for us. The idea is a p-value measures how likely the observed result occurs at a random chance. So the actuality of it is that if the p-value gives you 0.005, it means there's a 1/20th of a chance that what you're seeing occurred randomly, and if as you go lower, it increases the chance of that randomness happening. So this is the golden standard in science is 0.05. And to be honest, there's no real hard and fast logic as to why we selected 0.05. It's just a nice round number that generally works. So to be honest with you, a lot of science and significant findings are based off this seems good enough, which is a little bit scary to think about, but also means that a lot of the time, if something seems good enough to you, you can probably just go with it. So I have a question for you. Let's say, for instance, you had a family member who needed a very dangerous but life-saving drug treatment. And they said that the p-value chance of the effect actually happening was X. How low would that p-value chance have to be for you to feel comfortable with giving this loved one that very dangerous but possibly life-saving drug? Well, I might have... Oh, can you still answer? Can you guys still answer? Yes, okay. This one also doesn't count. I'm just trying to see where you guys are at, to be honest. Anybody else still putting down an answer? Interesting. So going with the scientific consensus, fair enough. I don't blame you. The question becomes, how low do you need to go? And that's something we sort of have to wrestle with. As you start producing your own scientific knowledge and new knowledge, you sort of have to come to your own consensus on how significant are my results to where I am confident enough to actually give them to the world. Interesting. So the take-home message from this little section on scientific philosophy is the difference between inductive and deductive reasoning, that whole idea of the inductive leap, because at some point we just have to make that step over. We can look at things until the cows come home, we can get as low as we like with our p-value, but we're never going to get to zero, unfortunately, for a lot of cases. And finally, what a p-value actually means. I found it very funny. I had a friend who went for a job interview and they just asked straight out, hey, what does a p-value actually represent? And a lot of people actually got it wrong. So now you won't, which is good. Moving on to the next section. Now there's a lot more questions in this section. So the competition's about to get a little bit more competitive. So it's more like a Kahoot style, so you got to answer with speed and accuracy. And obviously the best person with the score going 1 to 3 at the front. And these are the different data types we're going to cover today. So we're going to be covering whole genome sequencing, which is going to be very familiar to what stuff you've done in the lab. RNA sequencing, which is pretty much exactly what you've done in the lab. SNP array and variant data, very interesting, looking at different mutations within people. And finally, epigenetics and chip sequencing. Epigenetics are absolutely fascinating because it's actually heritable and you can change it. So if you are thinking about having a family anytime soon, get your epigenome as healthy as possible before you have a child because they will inherit it from you. We can go over that a little bit more though. Firstly, whole genome sequencing. The first step looks very familiar, DNA extraction as you've done in the lab itself. And then from that DNA, you're chopping it up with a bunch of enzymes into little fragments. And then once again, this also should seem quite familiar to you. We do a PCR on those fragments to get a whole bunch of them all together. And then finally, Illumina sequencing, something we've also done in the lab, where we take those fragments and we use fluorescence markers to see which bases go where. And then we take all those readings, put them in a computer, and the computer spits it back out into a long sequence for us. And the advantage of this stuff is we can look at various different structural changes. So we can actually, through comparing whole genome sequences to each other, see single nucleotide variants. We can see insertions and deletions. We can also see structural changes where bits of the chromosome have been swapped around. And finally, we can see copy number variations where genes are copied, etc. So that was a real quick rundown of whole genome sequencing, and you're going to be quizzed right now. You're going to get quizzed right away on it, so get ready. Here we go. Question 1. What is the first step in whole genome sequencing? DNA extraction, everybody got that one? Brilliant. All right, next question. There's multiple, here we go. Which of these cannot be analyzed through whole genome sequencing? Oh man. Which one? Oh, people are going. 3, 2, 1. Epigenetics, a lot of you got it. Well done. This is a problem. I think the quiz, hold on, I might have messed up the whole quiz. Anyway, we'll figure it out at the end. All right. PCR is non-essential to whole genome sequencing. And I misspelled false. This hopefully everybody got that one. There we go. Oh, man. Interesting. Here we go. Next, RNA sequencing. So this one should look very familiar to you because we've done it a whole lot within the lab. So first step, extracting the RNA. That we didn't get you to do. RNA extraction is actually really painful to do, very easy to contaminate. RNA degrades from anything. Because it's not meant to last a long time, because you have your DNA, and your DNA is the main copy, and your RNA is like the copy of your DNA, so that gets used up a lot. So RNA is actually not meant to last a long time. So RNA extraction, actually quite painful. But moving on from there, something we did get you to do was to convert the RNA into cDNA through reverse transcription, and for a 2-step real-time PCR. Hopefully that is sparking some memories for you all. Then we do the same thing again. We sequence them as we did before, same idea with this one. And then finally, we do something which is to build a count matrix, where we have all the different genes and then all the different samples going in a large matrix across each way. And we analyze it all through a bunch of different R packages, and I love R. RStudio is my mother language, and I love it. So, straight on into the next. No rest. Here we go. How do you build a count matrix? Count the genes. Count the sample. Count the genes per sample. Count the genes per sample. Lovely. Next question. What enzyme converts RNA to cDNA? Oh, here we go. Who remembers? Oh, one more person. There we go. Everyone. Lovely. What did we get? Yes, very good job. Everybody pretty much got the right one. Nice. And last question for this area. What is the molecule extract at the beginning of this process? And there's a color change too. Everyone voted happy. Very good. Nice one. Let's see, where are we at with the leaderboards? Oh, Metal, Lola, Lara, all doing very well for us as well. Oh man, a little bit of a shake-up. All right, here we go. So now we're moving on to the single nucleotide polymorphisms array and variant data. This one, very similar. Also can be considered to be used for a lot of patients. However, they start off with taking your blood saliva and getting your DNA extracted. We've heard that before. Then we take the DNA, we apply a fluorescence probe to it, and we run it over this chip, which has these these antibodies, which are specifically made for single nucleotide variants. So then it runs over this chip, it gets grabbed, and all of these DNA parts get stuck to a particular area of the chip and glow with the fluorescence. Then we use a computer to measure the different areas and the fluorescence in the area, and we know which single nucleotide variants has been extracted. So we use the different fluorescence. You can use different types of fluorescence on the same chip to get more results. And then from that, we know which type of single nucleotides you've got. And comparing it to whole genome sequencing, you can see that the cost is cheaper, but when you're doing an SNP array, it's only for already known single nucleotide polymorphisms. So you can't discover any new ones. And the data is only quite small, whereas the whole genome sequence is quite huge. However, the single nucleotide polymorphism is array for population genetics. So if you have a single person who's not part of a study, but just looking to see if they have a particular mutation, this is the way to do it. And if you aggregate the arrays aggregate, you can get about 500,000 to 5 million known single nucleotide variants in a single go, whereas the whole genome sequence gets you everything. So the main advantage of the whole genome of the single nucleotide array would be the cost, but as you can see, the whole genome sequence is actually not becoming that too much more costly. So maybe we might stop seeing single nucleotide polymorphism arrays, or maybe not. Maybe there's a different use for them. But you might have caught the trend by now. Let's go. What does DNA bind to on the chip? explicitly call them fixed probes, I'm sorry to say, but most of you got it right, so I'm sorry for those. I should have explicitly called them, they're called fixed probes. So sorry for a few of you that might have got mixed up there. That one's on me. How many significant variants can this technique detect? Oh, a few people might not get an answer. Oh man. I think I might have messed up this quiz. But the answer for this one was 500,000 to 5 million. Sorry about that. At least can this technique discover new SNPs? Oop, that one's also messed up. Yeah, I'm sorry, this whole quiz is completely messed up. I should have double checked these, I'm sorry. Well. Okay. All right. Epigenetics and chip sequencing. So this one is super interesting. So epigenetics is the idea of different molecules controlling the expression of your genes by either making it easier for genetic mechanisms to get in there, like your DNA polymerases, your transcription factors, or making it harder for them to get in there. And also, the idea is that these epigenetics can actually be transferred to your children. So if you're looking to have a kid, get as healthy as you can and get as good a mental as you can for like a year or two beforehand, so those can be passed on to your kid, and then you can sort of let it fall off. Yes? Oh, I'm sure it would have. Yeah. You know, I wouldn't be surprised if that's real. If your grandparents went through a large long-term famine, then their epigenetics would have been around conserving energy, and that would have been passed on to their kids if they had any. So this is a way where another way in which your parents can screw you over. So let's break cycles and such. Anyway, the first concept around epigenetics is DNA methylation, which is taking some methyl groups and applying them to cysteine and guanine islands, which is long stretches of rich cysteine-guanine base pairs. And what it does is it actually silences those genes, and it makes it harder for the histones to unwind the DNA and allow those different mechanisms to get into the genes and replicate them. So effectively silencing them. But this one is specific to DNA, so it binds to the DNA itself. The other options are a bit different as they target the histones. So histone acetylation. So acetyl group comes to the histones which house your DNA, and it loosens them out and makes it easier for different transcription factors and gene activating mechanisms to go in and replicate that section of DNA. Whereas histone methylation can be context dependent. It can either have the methyl group come and attach to the histone and either make it tighter and harder for replication to occur, or loosen it and make more occur. So there's two main ways that we can measure epigenetics. The first is called chip sequencing. And essentially what it does is you take your nucleus and you fix it. And what does it mean? It means when you're fixing your DNA sample, it locks all the proteins and all the transcription factors and all of that machinery onto the DNA. And then you have antibodies, which is the I in chip sequencing. And those antibodies come and grab onto those proteins of interest that are interacting with the DNA. And then, with those antibodies attached, you can separate those out from everything else. And you can then go ahead, release the DNA, and then go through the sequencing mechanisms we've talked about earlier. The other way to do it is known as ATAC sequencing, which is only specifically for those sections of DNA that are already open. So what happens is you have these TN5 molecules, and they go in and they find the open DNA and they separate them out and also apply adapters to each end of the DNA. And those adapters are what allow those open DNA sections to fall out of the histones and also allow them to be filtered out from everything else. And then you just go along and you sequence that DNA as we have with those other methods before. So two very interesting ways of how you can actually look at how open or closed the DNA is. The first one is using antibodies to immunoprecipitate out your proteins that are attached, or the sections of DNA that have proteins attached, which would indicate epigenetic activity. And the other way is to use the TN5 to just section out the parts of the DNA that are already open when you get there. And the epigenetics, another thing that's super interesting about epigenetics is it also controls pretty much everything else about your cells. Because your cells have all the same DNA, the epigenetics and the expression of those genes is what really determines if your cells become like a skin cell, liver cell, a hair cell, all the fun stuff. So epigenetics, super cool, inheritable, causes all your cells to be different from what they are. So I think super cool. Exactly. Exactly. It's all the way down. Speaking of all the way down, let's see if I didn't mess up any questions in this. DNA methylation and both silence and activate genes, silences genes, or activates genes. Can't seem to get a question. All right. Histone methylation. I think we should take a 5-minute break at this point. There's a lot's been covered. It'll also give me a chance to figure out what I'm going to do, because I think too many of the questions weren't quite set up correctly. But I'll have to honor it. We'll figure it out. All right, last section, here we go. We're talking about plots, and as you can guess, this one is a volcano plot, if you couldn't guess as to why. The fun thing about volcano plots is how good they are at showing you a whole bunch of data all at once. Don't worry if you're squinting your eyes, I'll show you what you're meant to do. So imagine, for instance, you've got two different cell types, right? You analyze the genes, Yeah, and then you have a whole list of genes, and you have the p-value for how significantly different they are between each other, and then the fold change difference. But how are you going to look at 1,000 different genes and their p-values and their fold change all at once? And in particular, pick out like the significant ones. It's going to be really tough. So what you might do is a little trick, if you're ever working with p-values, and you want to make them positive rather than get larger as they get more significant, is you take the negative log of your p-value and then it makes it larger as it gets smaller. Very handy if you want to represent it mathematically. And if you're doing that for your p-values, and then you also do the fold change and you take the log of your fold change, so the fold change meaning how different they are between the two different cell types. If you don't remember, sometimes the fold change can get really big, like in terms of thousands of fold change difference, and you might also have some fold changes being like down at 2, right? So to make that more easy to look at on a graph, you can take the log, which keeps it proportional in terms of how different it is. It just makes all the numbers smaller. So instead of it being like 1 to like 10,000, it might be 1 to 100 with still the proportional difference. So it's a lot easier to actually look at on a graph. So then you take your negative log of your p-value and you make it your x-axis. So as it's more significant, the sample gets higher. And then you take your log of your fold change, your log FC, and you make it your x-axis. Sorry, I might have said that the wrong way. Your y-axis. Thank you. So your negative p-value is your x-axis, your y-axis. So as it's more significant, it gets higher. And then your fold change, as they get either more negative or more positive, go further out towards the sides. And can anybody have a guess as to what that horizontal dotted line might mean? It's okay if you have no idea. What do you think of mine? Exactly. It is the line at which your sample is either significant or not significant. So then you can easily tell how many samples you've got in either direction, either increasing or decreasing, is significant. And that's why I love a volcano plot, because it lets you look at all these different genes, all these different samples, all at once, and is able to show how many are significant versus how many are just sort of middling or boring. Oh, baby. We've only got 3 more questions, don't worry. Is anybody else still looking to join? Anybody else? Everybody who wants to join is joined. All right. Sing out now if you're still wanting to join in. All right. I'll take that as we're ready to go. Here we go. The higher the y-axis, the blank the sample is. Thank goodness people were correcting me on my x and y sequences, otherwise this question would be a problem. Come on. There we go, more significant. All right, heat maps. Heat maps are also really cool because it's almost like a 3D effect, but on a 2D plane. You have, in this particular example here, we have our x-axis and our y-axis being different factors of a car, and then the color change being how linked they are in terms of power output. So very interesting in terms of another way to think about it. Imagine you have a whole bunch of correlations, like you're trying to correlate many different factors together. You're trying to see how tightly linked different factors are. You've got your p-value, you've got your R coefficient to represent how closely linked these are, either positive or negative. But imagine you've got a load of these and you're trying to look at all these different factors all at once. An easier way to do it would be to throw it into a heat map, where you have all your different factors, and then you put in your correlation for how tightly linked they are, and you give it a color for if it's closer to -1 or closer to +1. Heat maps are really good for this. I'm showing you different plots for showing a whole bunch of data at once, in case you haven't picked up on the theme. But heat maps don't just have to be used like this. They can also be used in many different ways as well. So for instance, if you're looking at the cost of rent and how it changes, you might have your different areas and then different years, and then the color might represent how much things cost. Or even the traffic intensity over different days, with one side being the day of the week and then the time, and the color being the number of cars or how critically overworked it is. Heat maps are very versatile and very useful at showing kind of like three-dimensional information almost. Here we go. Second to last, penultimate question. Here we go. What is the main advantage of using a heat map over simple table of numbers? There's a lot of reading. It automatically normalizes the scores for fair comparison. It makes it easier to visualize, detect patterns and clusters in the scores. It reduces the amount of data being shown, so there is less to analyze. Good work. Nice work, everybody. Last plot. I've saved arguably the best for last. This one might be a little bit of a brain tease, but I think you can get it. The principal component analysis. You don't have to understand the math behind this one, but the general concept about this graph is it shows how much variability there is in the data from one sample to another. So, in terms mathematically of what you might want to understand, you have all your different columns that relate to a different sample. So let's say, for instance, we're looking at the different variables of a person, such as their height, their arm span, leg span, maybe we go for BMI, weight, lung capacity, things like that. What the principal component analysis does is it looks for the variation between column 1, column 2, column 3, all the way down, and it finds out which columns have the most variance, and it squishes those down into a principal component 1 and a principal component 2. So the higher or the lower a principal component is, the more variation that sample has across all its data. And if the difference between principal component 1 and principal component 2 is the primary variation, the biggest part of that variance goes into principal component 1, and then the leftover variance goes into principal component 2. Don't worry about the math behind it. You get into vectors where we're talking about direction and size. Don't worry about that. You don't need to know about it. What you essentially need to know is how to read a principal component analysis plot. So think of it as each dot represents a different sample, and then the further apart those dots are, the more variation there is between those samples across all their data. Then what you can do is the closer a sample is to each other, the less variation is, or the more they overlap, the closer their data is to each other. And then what you can do is you can take all those different samples and you can color code them for the groups they're a part of. So in this example here, you have different types of cancers. So you have malignant cancers and you have benign cancers. And maybe we looked at the genes that are expressed in those. And what you can see is if we squish down all the variance, the variability in that data into the principal component 1 and principal component 2, we can see that some of the malignant samples go all the way to the left and up, and some of the benign samples go all the way to the right and down. So those samples are extremely different in their data. However, we also have some samples that are in the middle that are really overlapped and tightly correlated together, even though they're benign or they're malignant, which would indicate that those samples actually don't have much variation in their data. They're actually quite similar if we look at the data as a whole. But when we look at the group of malignant and the group of benign, we can see that as a group, they actually have two kind of clusters separate from each other and then a slim overlap together. So that's the important idea here of a PCA plot, is it shows how different samples are to each other or how similar they are. And when you color code them based on the groups they are linked to each other, you can see if the group itself is different from other groups. So it's a little bit of a mind bender, a little bit of a brain twister, but it's very handy to do a PCA plot early on to see if these different groups are similar to each other or different. Because imagine you do a drug test on some different cancer types and you see that you have this result here after exposing your cancers to this drug. You would say that the drug had an impact and caused the two groups to become different from each other, which would mean that there's something to look into. So, a lot to sort of digest in there. Don't worry about the math. Learn about how you can use a PCA plot to your advantage. You'll never have to calculate one yourself. There's plenty of tools online to do it for you. All right. Here we go. Last one. Here we go. What does the axis for the first principal component represent? The variability with the highest average value, the condition comparisons capturing the most variation in the data, the variable with the strongest correlation to response, or the axis with the fewest data points. Yeah, that one was right. Well done. Well done, everybody. Okay. Oh, here we go. And the winner is, who got there? All cake. Oh, Frosty got there at the end. Well done, Frosty. Who's Frosty? Who? Who's Frosty? Well done. Okay, so what am I going to... No, it does. So what I'm going to do is I'm going to honor the leaderboard. But I also have a box of favorites. So everybody can come grab as many chocolates from this box of favorites as you like. So, well done, Frosty. Amazing. Who's second? Who's second? Laura? Who's well done, Laura. Milk chocolate. Third, Agent X9. How did I know you were Agent X9? Ready, catch. All right. Everybody, please have some favorites before you go, as a thank you for turning up today. But that should be it, folks. Thank you for coming today. Yes. Yes. As a group, yes. In this example here, even though there is a little bit of an overlap, I would say as like a group, they do separate enough for there to be, it doesn't tell you what's different. It just, oh, thank you very much. It just tells you if there's a difference. Thank you very much.
And even, would that be overlapping in the middle?
Oh, yeah. Yes. Yes, yes. You